I am an architect by education and always had a very strong interest in biology, and for a long time couldn't decide what would be really my field, and was really happy to discover that there's this overlapping field of biomimicry and biomimetics when I did my PhD research, which feels ages ago, but in fact I did my PhD in 2008. And when I started, which was around 2004, it was really difficult finding an advisor connecting those fields of architecture and biology, and it's exciting to see that now the opportunities have exploded. There's programs, there's financed PhD in this field, and it really makes me happy to see that. And what I have prepared for today is a presentation of our most recent research project that we did at the University of Akron, where I'm based at currently, at the Biomimicry Research and Innovation Center. And the project is called Living Wall Systems Prototype, LIVAS. And it's a small research that we did in my lab together with grad students and undergraduate students. And it was about integrating systems or aspects of life into a prototype wall system and really put it out there, expose it to the elements, expose it to people who can visit it. And it basically is a very simple a wood construction that houses two research threads that we're following at the moment, which is mycelium material systems and leaf-shaped thermodynamics translated to ceramic tiles for facade systems. And when we started, or when we applied for the project, we thought about the objectives. So one of the main goals is to push this concept of living architecture further as an application field of biomimicry research and development, generating new design proposals for living wall systems, integrating new signs of life, for example, metabolic activity. And another real important objective was to really build a prototype and put it out there as a proof of concept, and also to show people what we're actually talking about. Many of the things that we're doing seem to be very abstract. And as soon as we put it out there with a prototype, it becomes tangible and people immediately grasp what we want to do. And finally, we also want to have some long-term monitoring of the stuff that we produce and work into furthering sustainability and integration of biology into architectural systems as well. So we started with a very brief design workshop where we very fast designed different concepts that were connecting to the aspects of life or signs of life as defined in biology. And we came up with about 40 concepts that we grasped in sketches and models, and we selected 18 of those further for drawing more refined plans and making more sketching and making some preliminary models. And those ranged, the content was really very wide, from aggregate structures that use self-organization for building systems to designing for integration of biology into wall systems and the like. And the whole frame of this was actually my PhD research on the science of life in architectural design that we took further and tried to find out what it actually means if we want to have a living wall. It would mean that it interacts with the environment. It would mean that there's probably a water cycle, maybe storage of rainwater, or maybe a slowing down of water by storing some water in the panels, creating maybe a vascular network. It's about thermal management, it's about material hierarchies, it's also still about generating space for life, it's about sustainability and allowing and designing for change. And so this was the outset of the workshop, and we managed to integrate some of those aspects into what we were doing. So what we finally created was this very simple structure to house those elements, and we decided to follow those two research threads that were actually there from previous projects that I did. For example, growing as building together with Barbara, who is in the audience and who will talk tomorrow. So this has a long history. And this is the kind of final image of the project that we were working on. And as I said, some of those aspects that we were talking about, which make up living architecture, were translated, others not, and we'll wait for the future and future research. So we produced those facade panels out of mycelium material. We experimented with different substrates, and I think most of you know about this material. So the mycelium is solidifying aggregate waste coming from agriculture that serves as kind of food for the fungus. And we worked here in collaboration with architect Chris Maurer from Red House Architects in Cleveland in his workshop facilities. And it was a real challenge to scale those mycelium productions up to that scale. It was not as easy as anticipated, so we had a lot of trouble growing this stuff, and we're relying a lot on previous expertise. And we were exploring also new shapes, or producing shapes by growing, optimizing the growth process, and taking on also ideas about, integrating the fruiting bodies and maybe the use of the fruiting bodies for the production of food. And we also had post-growth treatment of those panels. So we used some of the raw panels. Some of them were baked, so they were heated up to kill off the fungus. Some of them were compressed to half of the thickness. Some of them were baked and compressed. Some of them had an interior grid, a reinforcement material. And then we explored some various coatings that we applied mainly to purchased material from Ecovative that we also used in the installation. So our prototype had then finally two parts. So we had a site that we produced, and we had a site where we used Ecovative panels. And for that side, we applied the different coatings. And on our own production side, we explored the different post-growth treatments. And we left the panels on the installation since end of October, so within 3.5 months. They are still there, so they survived the Ohio winter. And we have some qualitative observations in the meantime. So for the baked panels, the texture became very soft. They started to disintegrate and almost become fluid and get large cracks, but they're still up there. The compressed panels were performing best, so they have a pretty hard texture, are structurally still good and just have small cracks. And the compressed and baked panels had a really big difference between the surface and the interior design, so the surface just came off really quickly and it looks very brittle. So we are looking at how those panels behave, and we hope to go further with this. The other part of the research was taking on my other PhD student who is not here, Ariana Rupp's research on plant leaf thermodynamics, and the observations that she made for plant shapes and how plant shapes have an influence to thermal behavior and also evaporation capacity of the leaves that allow the survival of the plants in a changing environment. And it seems that there is an inherent connection between the shape of those teeth and the evaporative capacity. And we explored that in preliminary biomimetic translations. And we observed the weight loss through evaporation and thermodynamics with a thermal camera. And we translated our preliminary findings to three-dimensional ceramic systems that we produced in a very traditional way. So Neil will laugh about this. And he actually gave some input on the clay composition when we were wondering what would be the best way to make those things. And we produced those panels at Myers School of Art at the ceramics department with a lot of support of the people there, and exposed those panels for a short term to the prototype. And we removed them because they would not withstand the frost, and we will continue our long-term observation now in spring as soon as the climate will allow us to continue. So the goal is to observe the thermal behavior of those different shapes, and it seems that the translation of those shapes is actually also successful in a three-dimensional setting, which was the big question. And so we will continue this long-term monitoring, and our plan is also to integrate a watering system and to have this water flow also present in the prototype. And finally, I want to thank the people who were well involved, which was mainly Thibaut Houet, my PhD student, who is here and actually presented the poster. Thibaut. He did the mycelium paneling together with Brian Foresi, who is a biomedical engineering student, and Ariana Rupp did the research on the ceramics part. And we were supported by the ceramics department, by Chris Maurer from Red House Architect, and by a few people who helped us put up the stuff, and by the biology field station manager, Lara. And so the prototype is out there, people can visit it, and people are going to the field station and to the park to have a walk. So this thing is really exposed to the general public, which is also a very, very nice aspect of it. And so this was just the most recent project that we're working on. We're working on other stuff as well in the lab. But due to the 10 minutes limitation, I think that's it, what we can talk about today. Thank you for your attention.
So the material on the surface, would that respond to the UV changes, and did it respond to, let's say, temperature changes during the arc of the day, or is it longer term kind of responses that it makes?
That's a really good question. Some of the panels were actually dead, so the ones that were baked did not contain live fungal material. The ones that were raw contained really the living system. And what was intriguing about the panels, and I didn't say this in the presentation, but they smelled really nicely. So when you passed the wall, the smell was just amazing. It was really this very fine smell of the fungi. And they had a lot of fruiting bodies out, so they also looked really amazing. That was another, aesthetic and sensual dimension of the project that is not so obvious maybe. And for the live material, for sure, the environmental exposure had influence on the organism. And for the dead panels, they are just disintegrating more or less slowly depending on the treatment. So the question is for us rather, does it withstand long enough so that we can use this material maybe as a growth substrate for green facades? Another interesting thing for us was, would it be settled on by other organisms, which we could not monitor so far? So during the Ohio winter, I think there's not a lot of chance for other organisms to settle, but now as spring is coming, so this becomes an interesting observation. And so that's actually something that we will monitor over the summer as well. So, in terms of temperature and humidity, yes, there's a lot of influence, and also the orientation of the wall was a big topic. So should we orient it to the south, or what do we do, or to the weather situation? So as we all know as architects, this has a really huge influence on what's happening. But for us, it's also important to really do outdoor exposure experiment and start somewhere, even if it's not, grasping all the aspects that we could research at once. Yeah. Does that answer your question? Thank you. Thank you.